Good morning, everybody. We will be starting in a couple minutes. We're just going to wait to admit people and then we'll start. Good morning, everybody. It's 9 o'clock. We have a lot of material to cover, so I'm going to go ahead and get started. Let's go ahead and advance the slides. You guys know who we are, most of you, so I'm not even going to cover this slide. This is our program overview. Next slide. Again, you guys have seen this before, and so I'm not going to go over it, and you can look at it after when we send out the slides. Same with this slide, except I want to point out that starting next week, we are going to be adding Mpox clade 1b to our list of pathogens we target, and we are sunsetting rotavirus. So what we are currently monitoring is changing a bit, and we want to make sure we have time for questions about that. So that's why we are sort of rushing through some of these repetitive slides. Next slide, please. And next slide. We have our open access protocols. We do have a protocol for Mpox clade 1b, which is not up here, which we have sent out to you via email, and we will add for the next slides that we send out. Next slide. We do want to point out some new recent papers that have come out of WastewaterSCAN. The first two are a paper. One, it was headed by our colleagues at CDPH, looking at estimating reproduction numbers for COVID-19 using WastewaterSCAN data. And then yesterday, we had a paper released on a study of the infectivity and persistence of influenza viruses in raw milk. And that is published in ES&T Letters. And you can access that through this live link when you are sent these slides. And then we have also a paper that was recently accepted where we collaborated with some data scientists at Carnegie Mellon to look at the variability of PMMOV biomarker in wastewater across the United States. And if you'd like to keep up with our published work, we do have that link, which you can access when you get the slides later. Next slide. We also developed a new hepatitis A fact sheet because we were getting a lot of questions from partners about how to interpret the hepatitis A data. And so you can also access that fact sheet anytime you want if you have questions on how to interpret the data. And it also has a link to our publication on hepatitis A in wastewater. Next slide. We also have a new section on the publication page, which is supporting data interpretation. Over the last few years, as you know, we've written up a lot of fact sheets and guides from everything on variability of measurements in wastewater to poliovirus to Mpox. And so we have put those on our website for easy access for us when we get questions from you all, and also as a resource for you to look at. Some of these were written, not so recently, but the information in them is still valid. Next slide. So an update on the H1 assay. As you know from your email, we resumed measurements around the beginning of December. The exact date at which samples started to be processed varied because we implemented it in the laboratory, and then samples come in from different times. As you might remember from previous work, this graph is some retrospective work we did last influenza season with H1, H3, and H5. And you can see how H1 in yellow was almost as high as the black general influenza gene. The H1 assay was working great and aligned with what we expected because we knew last season that H1N1 was the most common circulating subtype of influenza where these samples came from. And what happened is over the summer, there was increased efforts to test and sequence flu because of H5 circulation, and a bunch of new sequences were deposited in GISAID for H1N1. And it turned out that there was a mutation in the region of the HA gene that our old H1 assay targeted. So we are using a redesigned H1 assay. It is one that was provided by CDC's flu group, which is a great, wonderful researchers in the flu group. And we confirmed that the assay should work and worked. And we deployed it on November 29th. And just a note for all of us is that viruses like influenza A evolve quickly, especially in the region of the HA gene. And so we need to constantly keep up with checking assay performance for these. And this is something we'll have to do forever into the future. Next slide. So things we're thinking about for influenza A data interpretation right now is that, the influenza A M gene assay will detect M genes from the H5N1 virus as well as those from human influenza. So if we see increases or high levels of influenza A M gene, we don't know right now if it's from H5 or H1N1 or H3N2, the more human associated influenzas. And so, we have that data alone could be confounded by the presence of dairy products potentially in the waste stream. And so this could affect our interpretation of the data. And currently we use the M gene concentration to determine influenza A wastewater event onset and category. So that could be affected by the presence of H5 in some locations. So really, we're going to need to look at H1, H3 measurements, as well as H5 in some of these locations to parse that apart. And so we're working on that right now and thinking about the science and the data science to do that. We're not going to be making any changes in the near future, but we do want to alert you that if your wastewater treatment plant or in your region, if you're seeing notes that influenza A is high, wastewater category or in onset, that you should look closely at the H5 data to see if you have potentially any interference from inputs from dairy processing facilities. Now, we know that in most places, H5 concentrations are quite low, and it's only in pretty unique locations where they've been high, where you have actually a dairy processing facility that has inputs of waste that is generated using milk that had H5N1 genomes in it. So, okay, so more on that as it evolves. Next slide. I want to go over the H5 detections in wastewater we're seeing. So this is as of 3 days ago, what we have seen across the United States. The gray states are states where there has been at least one positive detection since we started in mid-May 2024. And red indicates a wastewater treatment plant that has had at least one positive detection. And so you can see there are a whole bunch of plants that have never had a detection. But there are plants that have, and I think most recently we've been watching in awe, if that's the right word, as all of California's wastewater treatment plants have lit up like a Christmas tree. So it's not funny, but you gotta laugh when you can, or a little like smile when you can. So our most recent detections are in California, Florida, Delaware, Iowa, New Jersey, Tennessee, Texas, and Utah. And in most cases, the concentrations that we're observing are low, but definitely positive. Next slide. And then here's just to show you a little bit about the types of concentrations that we've been seeing. And actually, I can't see the bottom. Oh, there we go. So this is just to point out to you, if we look at a histogram here on the x-axis is H5 copies per gram in wastewater, and then the number of samples that have been positive in different categories. And what you can see, as I was mentioning earlier, is that most of the measurements that we're seeing are in this 1 to 5,000 copies per gram in wastewater, which is low because our detection limit is about 1,000. Like we can detect things less than 1,000. That's why there's a less than 1,000 here. Those are actual positive measurements that are less than 1,000 and truly positive. But most of the measurements are very, very low. meaning it's unlikely that they can contribute to like an upward trend in wastewater. And then we have some measurements that are higher, and the ones that are very, very high, like greater than 500,000 copies per gram, those are in locations that usually have like a milk processing facility that is releasing treated milk or milk waste into the sewer system that has the genetic markers for H5. Okay, next slide. This is, I think, the last slide that I'm presenting today, and then I'm going to pass it off to Marlene, and then Marlene will pass it to Amanda. But I wanted to briefly describe the results of our research on infectivity and persistence of influenza A in raw milk. So what we did is that we collected raw milk from an actual farm in California. We seeded it with H1N1, which is a surrogate for H5N1. And there's actually been some study, actually one of Marlene's colleagues at Emory has done some work showing that H5N1 and H1N1 decay similarly in milk that's on surfaces. And they also decay similarly based on a systematic review that my group has done in water. So it's a reasonable surrogate. We seeded it into raw milk. We calculated a first order decay constant for the infectious influenza, and the T99, the time for 99% decay of the influenza we seeded, was 3 days, meaning that the infectious influenza decays over time, first order. But we did not see any decay of the influenza RNA target over the 57 days that we stored the raw milk. Now, it smelled really bad after 57 days, but the nucleic acids were still there. We also tested the effect of pasteurization. As expected, it destroyed all the infectious virus. We cultured the virus with canine cells using a standard approach, but the pasteurization process only reduced the influenza RNA by 1 order of magnitude, sort of documenting scientifically what we already know is that pasteurization does not destroy the nucleic acid target that we look for influenza. And so you can read more about the paper. I think Amanda put it in the chat, but it just was released yesterday. Next slide. Okay. Finally, wanted to remind you, we do have this fact sheet for interpreting wastewater concentrations of the H5 marker, which you can access through this link, and also your local health departments and state health departments may also have fact sheets. I know there's one in California and potentially in other states as well that you can access for help in interpreting these data, or also reach out to us. We are always available to answer questions, and we have answered a lot of questions this past week as well. Next slide. Okay, Marlene, it's you.
Okay, we're going to take it in a completely different direction now and talk about Mpox Clade 1B. Okay, so you should have received an email from us about this, but we want to share some of the information about what we're planning for testing for Mpox Clade 1 and specifically Clade 1B in wastewater. So this is the assay that we've chosen to use. This table shows the primers and probes, and you can see the reference here for the paper that reports on this assay and validates it. So we're using, we're implementing the Mpox Clade 1B testing using this assay, and we have a methods document that we're sharing with everyone that we has all of the details of exactly how we're implementing that. You're right, it will not include Clade 1A, so it's not pan Clade 1. It is specific to Clade 1B, which is the currently circulating outbreak clade of concern for Clade 1. This is the workflow that we typically follow for scaling assays and that we are following in this case. So we have been working with a subset of plants for about 2 weeks using this Mpox Clade 1B assay. All of the results so far have been non-detect. We have a staging site that we have started to pull that data into, and it's working smoothly. So everything seems to be on track. We've gone through this process of the decision to track Clade 1, which has been a high priority, we know, for a lot of our partners, and is a high priority for CDC. Choosing this assay, doing testing to make sure that it is specific and sensitive. We have some of the information reported on that in the methods document that we've shared. Then we've gotten the materials and now done this kind of 2-week pilot. So we're ready to deploy the Clade 1 assay more widely to the entire WastewaterSCAN network. We'll be doing that still on the staged site, so this data won't be going on the public dashboard quite yet, but our plan is to start the testing for Clade 1B at all WastewaterSCAN sites on this Monday, December 16th. As Ali mentioned at the beginning of the call, in order to do that, we'll be sunsetting, I think this says unsetting, we're sunsetting the rotavirus assay. So we will not have that data stream anymore, but we will have Clade 1B data coming in starting next week. Again, that will be sort of the lab will start to process samples on December 16th. So whichever samples are being processed on that day, depending on the date of collection, that's when it will start for your site. This is the timeline of all of the details you need to know. So December 16th is when we'll start that prospective Clade 1B testing at all the sites. We will have, again, the data available on a staging site. We're not going to make any changes, especially during the holidays. I hope some of you are taking some time off and relaxing. And so we don't want to, have anything changing drastically during that time period when people have more limited availability. So the data will be available to you. We will send you the link for the staging site. As always with our staging sites, that will be a link that's only distributed to our partners. However, it is not password protected. It is a live link. And so, we ask you to kind of keep that among the group. And then in early January, assuming that things are going well and we're confident that everything is in place as it should be, we will put that staging site data onto the public dashboard. And then at that same time, we will start working on the submission of Clade 1B data to CDC News. So we will have the data available starting next week, but we won't be sharing it publicly or on the dashboard until early January. We put together this Clade 1B FAQ document. So a lot of answers to commonly asked questions. Let us know if you have additional things you want us to put in here or address in this document, because this is a living document and we're happy to add to it. And then we also have methods a methods document, as I mentioned, that includes some of that sensitivity and specificity testing, more details about the assay. And so we'll share that as well. Okay, and then last thing before I pass it off to Amanda is ongoing research projects. So we just wanted to update you and mention a few of the ongoing research projects that we have that are not part of what we're scaling at this point. So we are working with several partners on parvovirus. So that paper is almost done. Several WastewaterSCAN people are working hard on getting that done soon. So that analysis is nearly complete. And then we are working on Bordetella pertussis. So we know that pertussis has been of a lot of interest to a lot of people. We're in the earlier stages of working on pertussis. So there's quite a bit more research and development we have to do on that project, whereas parvovirus, we're sort of nearing completion to publish that work. And then antimicrobial resistance genes, we've shared with you in previous calls some of the details of this analysis, and you can see some of the preliminary results here, looking at the distribution of ARGs at our sites across the United States in cross-sectional samples from just a short time period earlier this year. So you can always go back and also watch the recording for that October 2024 call where we talked about that study in more detail. And I'll pass it off to Amanda.
Thank you. I'm going to go pretty quick through this, everybody. We're going to send out the slides afterwards that have a lot of information, but we want to leave plenty of time for questions at the end if there is any questions about H5 or Mpox. So just as a reminder, we use a categorization method that is based on levels, trends, and frequency. And Stephen just put the methodology in the chat for you to refer to. Next slide. And as a reminder, we have 3 archetypes. As we are sunsetting rotavirus, that'll no longer be generating categories, and we are not currently calculating categories for H1, H3, and H5. Next slide. Next slide. So high-level summary, SARS-CoV-2 is in the high category due to an upward trend in median concentrations. There's currently a 96% detection rate for SARS-CoV-2. Influenza A and influenza B are currently not in seasonal onset, although we are seeing more consistent positive detections across the board. RSV recently went into seasonal onset, and it is in the high wastewater category. I'll go into that a little bit more in the next slide. Human metapneumovirus is in the low wastewater category, and that is due to only a 20% detection rate at this point in time. EVD 68 is in the low category. Before last week, EVD 68 was in the high category and was in seasonal onset. So we've seen concentrations come down pretty significantly from the peak that we observed in September and August earlier this year. Neurovirus is in the high category with 100% detection rate. 73% of sites are in the high category as well. Rotavirus has a 66% detection rate currently. Mpox, Clade 2 are all non-detect in the last 21 days. C. auris, we've had only 8 positive detections in the last 21 days. And Hep A, we've had a 13% detection rate in the last 21 days. Just as a reminder, I know that was very quick, but we will be sending out the slides afterwards with more information and maps and some of the counts and categories as well. Next slide. So as I mentioned, I'm going to go into RSV a little bit more. We're kind of at the start of RSV season, and we're currently in the high wastewater category. So these two plots on the left, you see the national aggregated line for all of the WastewaterSCAN sites. And then on the right, you see the current positivity rates with data pulled from the CDC yesterday. Currently, we have a 79% detection rate in our samples, and RSV just went into seasonal onset earlier this week, I believe on Tuesday. So we are really starting to track this and see concentrations across the board increase across the entire WastewaterSCAN network. And here's just a quick map. As you can see, the west, the south, and the northeast are in that high wastewater category. And I think that that's it because we want to leave plenty of time for questions. So you can advance, Marlene. So just as a reminder, we have this general email account that goes to Allie, Marlene, Stephen, and I. So if you have any questions about the data, please feel free to email that. Next slide. And typically we send our newsletters at the end of the month, but due to the holidays, I will be drafting that and sending that off to you all next week. And our next meeting will be early January. So I think we are done presenting. So if anybody has any questions.
Yeah, we wanted to make sure we left time for questions. So please, here we have one in the chat. Will the Mpox staging site leak be shared with wastewater operators too, or just the public health officials? We are planning to share the stage link with all of our partners, which would include the operators as well as the public health officials. I'm glad you like the raw milk paper, Lin. Thank you. We're going to stop the recording.